Hello, everyone. Welcome back to the EDC Symposium. We'll now start our panel on work across the government. Our first panelist is Catherine Obie. Catherine Obie is the Senior Advisor for the U.S. Environmental Protection Agency's Endocrine Disruptor Screening Program, EDSP, and serves as U.S. Head of Delegation to the OECD Working Party on Pesticides. As Senior Advisor for EDSP, she leads the agency's regulatory implementation of endocrine screening within pesticide assessment and licensing decisions. Beyond EDSP, Ms. Obie has led the advancement of protections for human health and the environment in a variety of roles, including as Senior Scientist in EPA's Environmental Fate and Effects Division, Associate Director of the Conventional Pesticide Chemicals Registration Division, and Acting Deputy Director of the Office of Wetlands, Oceans, and Watershed. Catherine, whenever you're ready.
Thank you so much for the introduction and for the chance to be here with you all today. It's really an honor to be among these phenomenal panelists. If we were all in the same room, I would ask you all to give them a round of applause right now. I guess you can hit the reaction button on your Zoom if you feel moved, but it really has been a tremendous series of presentations already these last 2 days, and I look forward to the rest of the day. I'm going to move rather quickly through my slides so that my colleagues here on the government panel also have ample time. But please do put any questions in the chat. I think we'll have some time at the end of the presentations to respond. And you'll also see my email address on the last slide, so please feel free to make note of that. We've heard a lot these last 2 days about the importance of understanding the complexities of endocrine disruption and its larger implications for women's health in particular. Today, I'm going to share with you an overview of US EPA's work in this space, particularly through our endocrine disruptor screening program. EPA, or the Environmental Protection Agency, is a science-based agency whose mission is to protect human health and the environment. The last statistic that I saw of all federal agencies, only NASA, I think, employs more scientists than EPA. The endocrine disruptor screening program is housed within our Office of Pesticide Programs, which has a robust infrastructure for risk-based evaluation and regulation of all pesticides that are manufactured, used, or imported into the United States. Numerous statutes govern the regulation of pesticides in the U.S. You can see a few of them here. These laws require that each pesticide be registered to ensure that it is safe to use as directed, so the label is the law, and that it will not cause unreasonable adverse effects to wildlife. In 1996, the Food Quality Protection Act further required EPA to screen pesticide chemicals for potential estrogenic effects in humans. EPA also has additional discretionary authority to consider other types of effects, such as androgen and thyroid, consider effects in wildlife, and to screen certain substances beyond pesticides, such as those that are regulated under the Safe Drinking Water Act. EPA uses the World Health Organization definition of endocrine disruption. That definition includes consideration for possible adverse effects in the whole organism, its offspring, or population level effects, which occur as a consequence of altered endocrine function. And we developed a two-tiered screening and testing regime to try to get at both parts of that question. As you see here, Tier 1 was envisioned as a battery of both in vitro and in vivo screens, so sort of test tube and whole organism studies, to look for possible interaction with estrogen, androgen, thyroid, or steroidogenesis pathways in humans or wildlife. Based on the data from these chemical-specific screens and any other scientifically relevant information, our scientists make a recommendation on whether additional data on adverse effects are needed to inform the risk assessment for that chemical. In other words, are there gaps in our understanding? If so, we can require the submission of additional studies, which could include one or more of the Tier 2 guidelines you see here, or could be more targeted to answer a specific question. We initially used a list-based approach to identify which pesticide chemicals would go through the EDSP process first. These lists did not signify that the chemicals were likely endocrine disruptors, but rather that there was high exposure potential. So it was a subset of the universe. We shared all of the EDSP test guidelines for public comment, and we took both the guidelines and our weight of evidence review process through external peer review with multiple FIFRA scientific advisory committees. So that was a start, but remember that list one, as you saw on that slide, funneled down to testing and analysis for 52 pesticide active ingredients. We have more than 1,200 pesticide active ingredients registered in the United States, with that number growing every year. So there is a problem of scale here. If we expand to further consider other discretionary authorities, the universe of chemicals subject to endocrine screening grows considerably larger. So there is great interest in leveraging scientific advances and other efficiencies to better prioritize chemicals for screening, more rapidly screen them, and possibly to serve as alternatives to the more time and animal-intensive Tier 1 and Tier 2 tests. Enter new approach methods. We are very grateful for productive collaborations with our own EPA Office of Research and Development, the National Toxicology Program's NICEDM, international partners, and others who continually push the science forward. You can see examples here of some of those products, such as high-throughput assays and other tools that are in different stages of development. In January of this year, we published a draft paper to the public on the availability of different new approach methods that could be used for the endocrine disruptor screening program. While I don't have time to go into the models in detail here today, I can share that that paper concluded that the high throughput estrogen receptor pathway model, which is based on a battery of 18 in vitro assays within the existing ToxCast program, could be used as an alternative for 3 of the current Tier 1 validated assays. Similarly, we concluded that the AR binding model, which is based on a battery of 11 in vitro ToxCast assays, could be used as an alternative for the AR binding in vitro assay. Briefly, these in vitro models use high throughput technologies. In other words, they test multiple chemicals at once to interrogate key nodes along an endocrine pathway, building confidence on whether we might expect to see equivalent endocrine interaction in the relevant Tier 1 guideline assays. No assay is perfect, but taken together, these tools provide valuable information that can be obtained much more efficiently than testing one chemical at a time. These models and the reference chemical findings were taken to our FIFRA scientific advisory panel for peer review in 2017. And if you're really interested, you can Google that and find some of the documentation. On this slide, you can see the AR full pathway model. And work continues to develop and validate tools for other pathways, including steroidogenesis and thyroid pathways. In addition, EPA proposed that a number of new approach methods could potentially be used now to support priority setting and as other scientifically relevant information in weight of evidence determinations. You can see examples here, including reduced subsets of those high throughput pathway models, quantitative structure-activity relationship models, and other tools that integrate and extrapolate information across and from multiple sources. This is an area where the science is rapidly evolving, and what is state of the science today might not even be available in 5 years. Another consideration is that these assays can be expensive. And so we are exploring whether reduced subsets of the ER and AR pathway models can provide equivalent performance to the full pathway models, and thus also serve as alternatives to some of those Tier 1 guideline studies. An example here of the reduced ER pathway model. This focuses on those key steps: binding, dimerization, transactivation, and proliferation. And you can see that this model considers both antagonism with the nodes in red and agonism with the nodes in blue. EPA established the Endocrine Disruptor Science Policy Council, which we fondly call EDSPOC, in 2022 to advise in part on the use of new approach methods in EDSP. This internal multidisciplinary committee also reviews proposed EDSP exemptions and the use of other scientifically relevant information in EDSP weight of evidence determinations. Under the law, EPA can exempt a chemical from EDSP given sufficient information to conclude that the chemical is anticipated not to produce an effect in humans or other organisms, similar to an effect produced by a naturally occurring estrogen, androgen, or thyroid hormone. You can see on this slide some examples of exemptions which have been issued to date. The establishment of EDSPOC was a key milestone in ensuring continued progress in EDSP. Beyond the science, we are strengthening our organizational infrastructure to ensure that we are taking a thoughtful, durable, and transparent approach as we move into the future. And so I'll end here with a snapshot of just some of our near-term milestones for the program. These are really just steps along the path. They're not outcomes. They're part of the big picture where we are actively working to define implementation strategies and meaningful measures that make sense today and take into account advances in the science and other programmatic changes since FQPA's enactment and their original vision for EDSP. So earlier today, I heard a great question about overcoming apathy on the topic of endocrine disruption. For our part, I can share that it is a high priority for EPA to make visible progress in both the science and the implementation of endocrine screening. We cannot do that alone, so I especially appreciate the chance to be here with all of you and share with you today. Thank you for your time, and I think we'll hold questions until the last presenter.
Yes, thank you, Catherine. We will hold questions till the end, correct? But please feel free to put questions in the chat as we go along. And thank you so much for your presentation, Catherine. It's great to see the work that the EPA is doing. Our next presenter is Dr. Suzanne Fenton, who goes by Sue. She is a scientist leading the reproductive endocrinology group and the mechanistic toxicology branch at the National Institute of Environmental Health Sciences. She previously led work in similar areas at EPA, totaling 25 years of research on environmental factors affecting women's health hazard identification. Her laboratory has characterized chemicals or environmental factors contributing to mammary gland developmental defects and cancer susceptibility, puberty timing, pregnancy-related disease, and persistent adverse health effects in developmentally exposed rodent offspring. She has received several NIH and EPA-based awards for her research on perfluorinated chemicals and endocrine disruptors. Dr. Sue, whenever you're ready.
Thank you so much. Thank you for the invitation today. And I am making this presentation at the request of Dr. Wycheck, who is not able to be here today, and he apologizes. So the NIEHS has long been involved in the area of EDCs and women's health, and the our location is in Research Triangle Park, not in the DC area or Bethesda, and we are part of a larger DHHS, as you can see here. The NIEHS aims to provide global leadership for innovation and innovative research that improves public health by preventing disease and disability. And we have 3 divisions. that all have ongoing activities in the EDC and women's health arena. One is the grants that many of you receive from the Division of Extramural Research and Training. Another one is in our Division of Intramural Research. And I am actually located in the Division of Translational Toxicology, which used to be the DNTP. We have lab space and contracted research in that group. The much of the early work that went on in the area of environmental estrogens was introduced yesterday by Dr. Andrea Gore. We have like a 50-year history now of studying environmental and pharmaceutical chemicals that have developmental origins and adverse adult health outcomes. DES is just one example of that, which was introduced yesterday. And Dr. McLaughlin and Ken Korak, Retha Newbold had numerous publications in the area that really spurred some of the early work in the area of endocrine disruption. I gave a presentation at the Endocrine Society meeting in 2005, which resulted in a well-cited paper in endocrinology in 2006, proposing that these windows of susceptibility exist. And honestly, my presentation met with some skepticism. So more recently, most of us here especially have come to appreciate how chemical exposures at an early age, when organ systems are developing, can lead to health effects in childhood, puberty, and even into adult life. NIEHS has long supported the developmental origins of health and disease research. These windows of susceptibility are mentioned part of the RFPs in many of the actual programs in our system. And clearly the prenatal period is a sensitive window for lifelong health of an individual, and we have a robust grant portfolio supporting this concept. You heard from several NIEHS extramural grantees on how they have been studying environmental exposures affecting women's health endpoints. And some of those areas are mentioned here. And these one older project area that wasn't really mentioned is the BSERP, which is the Breast Cancer and Environmental Research Program. It did have some DOHAD approach in it, but it also evaluated multiple endocrine disruptors, looked at puberty timing, mammographic density, all and how they influence breast outcomes. There are hundreds of publications from this really productive area that really change the way we think now about how the environment affects disease risk for one of these really important diseases in women. So also noted by our speakers yesterday and today, the work that's been going on in the DOHAD research area over the past couple of decades was really spurred by Dr. Jerry Heindel, who's now retired, but you wouldn't know he's retired. He still has a lot going on. But, you know, Thad Shug has really taken over the reins of that program. And today I'm going to just talk a little bit more about some of the current DOHAD initiatives and then some of the internal projects going on in this area. So you have heard about some of the projects looking at development of obesity, metabolic syndrome, transgenerational inheritance, role of microbiome, and more recently, this focus on the placenta in the early origins of disease program, the maternal health program, and the children's environmental health programs, which Kim Gray holds a pretty significant role in. The areas shown, the grant programs that are shown here are really making an impact on the health and wellness of women and children. And so the EPOD program has been going on since 2014, looks to really better understand the role of the placenta in maternal and fetal health. And the preconception exposure program is contains work that's established in animal models, looks at pre-fertilization exposures, and has a really comprehensive like mechanistic analysis of how environmental endocrine disruptors may affect outcomes in the first generation offspring. And then also the pregnancy as a vulnerable time period of women's health is a more recent program headed by A.B. Boyles, and has several projects funded that are focused on endocrine disruptors and not only just birth outcomes, but later time periods in a woman's life and her health in general. Also, in our intramural research program, you heard yesterday from Franco De Maio and Corwin Williams, but there's also other work going on in that group internally in NIEHS, looking specifically at fertility, how the environment and endocrine disruptors may affect fertility, development, sexual development, and the placenta and how it may affect birth outcomes. And then in our epidemiology branch, there's also work ongoing in the sister study, which many of you I'm sure have heard about. It enrolled over 50,000 women who have a sister with breast cancer between 2004 and 2009. And it has several co-PIs shown here. And they are looking at numerous health outcomes and exposures that are including endocrine disruptors. One example that I'd really like to highlight now is a recent publication by Alexandra White on hair straighteners and relaxers and uterine cancer incidence. So there was more than a 10-year follow-up period here, and the hypothesis was that chemical hair products could be associated with higher uterine cancer incidence. And these products had previously been associated with breast and ovarian cancer incidence in this study. And in this follow-up study with almost 11 years follow-up on average, she did report hair straighteners having a significant increase, twice as likely to develop uterine cancer in this study. And you can see on the bottom there the paper if you'd like to look at it further. Also within the DIR, we have really stepped up our game in terms of the social and environmental determinants of health initiatives and programs focused on health disparities, really with a U4 focus. So, focusing in on underrepresented, understudied, underreported, underrepresented, and underserved communities. And so, we have developed an EA, Environmental Health Disparities, Environmental Justice faculty, which has quite a few members in it, I'm one of the members. We've hosted numerous really well-attended workshops focused on environmental impacts and really involving communities in these workshops. In addition, Joan Packenham, there's a lot of people that are involved in this, and I won't highlight them all, but Joan Packenham has run this women's health awareness program where we really have really large community engagement. It's been super well attended, and it continues to draw a lot of attention. And then there are two new research, funding programs shown here that one of which will have regular due dates, and one just closed in February of this year. But these are really focused ways in which we hope to bring more attention to environmental determinants of health for these communities. In the Division of Translational Toxicology, we have a really a nice pipeline that kind of represents all the different capabilities and tools that we bring to bear on how endocrine disruptors may be affecting women's health. Some examples of some of the work that's going on there are in our health effects innovation program, the cardiovascular effects. Brandy Beverly is leading a project on a scoping review of biomarkers and hypertensive disease program. And then another highlight is work that's being done on botanicals, which are found in are not regulated, and so our consumer products testing and our combined exposures and mixtures groups, led by Cynthia Ryder and Vicki Sutherland, are looking at the effects of different botanicals. Also, in our responsive research programs, both the sustainable alternatives and the emerging contaminants, we've been looking at bisphenols and PFAS compounds and how they affect pregnancy outcomes. And Darlene Dixon has been researching fibroids and some of the mechanisms of how environmental contaminants and endocrine disruptors in particular affect fibroid development, especially as it pertains to women of color. And then last but not least, we have also been working on or contributing to the vast amount of literature now on how in utero exposure to PFAS may set up a lifetime of increased disease susceptibility. So Kelly Ferguson in the epidemiology branch of DIR has published work on the effects on hypertensive disorders of pregnancy. We are using animal models to look at adverse birth outcomes and other early life exposure effects on lactation and preeclampsia, and how that affects low birth weight and development of the offspring over time, and recently reported on metabolic disorders. And then Chad Blystone has also contributed significantly to the evidence of PFAS contributing to cholesterol and triglyceride elevation and thyroid hormone disruption over the lifetime, and has run 2 cancer bioassay on PFOA. So this is really a very quick overview of the wealth of information, both funded by and being reported by folks at the NIEHS. And I know we're going to hold questions till later, but thank you for your attention.
Thank you so much, Dr. Sue. It's a great presentation to know what's happening at NIEHS. We really love knowing what's happening across the government, and this is a good place to start. Our next presenter is Dr. Tucker Patterson. Dr. Patterson was selected as Center Director at the FDA's National Center for Toxicological Research, or NCTR, in March 2023, after serving as the Acting Director since March 2022. Dr. Patterson previously served as the Deputy Director for Research in the Office of the Center Director/Office of Research, a position held since December 2020. Dr. Patterson earned a BS in chemistry from the University of Arkansas at Fayetteville and a Ph.D. in pharmacology from the University of South Carolina. Dr. Patterson worked for 3 years as a toxicologist for the state of Arkansas at the Livestock and Poultry Commission prior to returning to NCTR in 2001, where he worked in the Division of Neurotoxicology as a senior scientist until 2010. Dr. Patterson has been involved in neuroscience and neurotoxicology research for more than 30 years and has authored or co-authored over 100 peer-reviewed scientific articles and book chapters. Take it away.
Thank you so much, Maggie, for that introduction. As Maggie said, I'm at the National Center for Toxicological Research, and I do want to point out that we have been here a little over 50 years now. We are part of the Food and Drug Administration. With our research facility, when it was established solely to perform toxicological research. And so we do not have a regulatory function, even though we are part of the FDA. And as Dr. Fenton pointed out about the NIHS, we are located here in the piney woods of Arkansas. So we're not in the metropolitan area there of DC. And so we have an excellent facility for research. And again, we've conducted research here for over 50 years now. I'm going to really give you a high-level overview of some of the projects that we have going on in this space right now of endocrine disruptors. But also, I want to take you back in time and show you a little bit of a timeline in some of the projects that we've conducted here. Currently, we have a perinatal health center of excellence that is managed within the FDA and managed out of our office here at NCTR. We also have an FDA Office of Women's Health that we work closely with on various projects. And so I just want to walk you through some of the things that we have going on and some of the areas that we think we're headed. So the endocrine disruptor knowledge base is one of the databases that was developed here within our division of bioinformatics and biostatistics. Of course, I saw where Catherine mentioned about moving into NAMs and using more in silico type modeling areas. And of course, we have been involved in that as well here at NCTR, moving away from the classic in vivo type studies. But if you want to look this up, I mean, if you Google EDKB, it'll be the first thing that pops up on the list when you do a Google search. But this particular database, very similar to our liver tox database that we've developed, our drug-induced liver injury ranked data set that we've developed. But what we're doing here with this particular database is doing predictive binding here. We know, everybody knows now, hopefully after over a day and a half of this symposium, what an endocrine disruptor is, that they interfere with the system, that they bind to receptors such as the estrogen and androgen receptors. And so what this particular database will allow you to do is look at the binding or predict the binding of any particular compound. And so we're moving towards where you can put a structure of interest into the database, and it will show you the binding affinity for the estrogen or the androgen receptors. This is what the interface looks like. You'll get an Excel spreadsheet that drops out, but right now you can put in a CAS number and upload your chemical of interest, and it'll show you the activity profile. Another database that we are working on is this estrogenic activity database, again, with our division of bioinformatics and biostatistics. And this has 18,000 estrogenic data points that we were looking at over 8,000 compounds. We have, you see, 4 different types of data there. And each of these, EADB has a set of database functions for searching chemicals and estrogenic activity data, browsing the database, exporting the data. So again, these are all on a public-facing website, so they're free to use, free to access. So I want to switch gears here, and of course, Dr. Fenton talked about the NTP, and we have worked closely really since the 70s with the National Toxicology Program, and our relationship with NIHS has been very fruitful. And I hope to show you some of that, that you'll get an idea of how much work has been done in this area. But of course, this isn't a new area of work. I mean, we think maybe endocrine disruptors are new, but we've seen how far back it goes. And so we started these studies in 1997 to address certain aspects of the emerging endocrine disruptor hypothesis. And we wanted to look at a broad range of endpoints, look at long-term consequences, so do more of a chronic type exposures out over the years, and just do a lot of curation in this area. So we picked a couple of compounds back in those days, genistein and ethanyl estradiol, or EE2. We knew that they were both potent compounds, and so we did a battery of studies there. You can see pharmacokinetic as well as neurotox is kind of where I came in, my area of expertise. Behavioral work, immunotox studies, and in addition to all these multi-generational and chronic toxicity studies. We had some other compounds that we examined as well, but just not evaluated to the extent as we did with these 2 particular compounds. And you can see, again, talking about the fruitful collaboration through the National Toxicology Program, that we're only showing here there's about 30 publications. If you go to that link, I'm only showing some of the high level publications here, but we did show that, of course, genistein and E2, they produce serum levels that were within the human range. They both induced dose-dependent effects consistent with estrogenic activity. With our genistein studies, of course, the females in the highest doses tested, we saw, effects across the board with that, reduced body weights, other altering the estrous cycle, and then across generations that continuously ingested genistein, and also similar effects with the EE2 compounds. So now let's get into shifting gears again and getting into all of our favorite endocrine disruptor here, bisphenol A. We know that it's a high production volume chemical, but of course, as Dr. Transandi pointed out earlier, bisphenol A is just one in the family of bisphenols that are contained in a plethora of products you see here with the linings inside of our food containers and our plastic bottles and our CDs. But I do want to point out that, currently FDA's assessment is that BPA is safe at the current levels occurring in foods. The issue is that you've got an ubiquitous compound here that, that is found just across the board in a lot of different products. So when you're going to do a study on assessing, toxicity or potential toxicity of something like this, you have to have an environment that is very controlled. And so we identified, data gaps here that existed, and that's really what NCTR's role has been within the agency since its inception. We want to see these data gaps that exist. We want to try to address these data gaps. And so we conducted a series of studies. We did toxicokinetics in multiple species over wide dose ranges. Our design included perinatal exposure and looking at internal dosimetry measurements there with BPA. When you think about BPA being in plastics and being, in other, compounds and other things that we're exposed to, when you're trying to do a study on BPA, you have to make sure that, the water's BPA free, that the caging is BPA free, that the bedding is BPA free. So the only BPA that actually the animal is being exposed to is what you're administering to the animal when you're doing a tox study. So this is the program on BPA that we first started with a toxicokinetic study in multiple species. We used that data and moved it over to PBPK modeling, where we extrapolated to humans. We looked across the routes of exposure, across life stages. And then a lot of people probably on this, that are participating in this symposium are very familiar with the Clarity BPA and the consortium there, which was just a huge endeavor. Multiple locations that were looking at BPA and BPA exposure and its effects on the various species that were being examined. So this is, again, just a snapshot of the peer-reviewed publications that came out of these toxicokinetic studies and these PBPK models. You can see, again, dating back into the mid-2000s. We had a 90-day chronic study, and we also had a 2-year chronic study as well, and that was really the core study of the Clarity BPA study. And again, you see the publications that came out of the 90-day study and the Clarity PPA core study, and I'm showing you the two NTP research reports that you can search these online and look at the comprehensive data that we came up with in these studies. The overall assessment there is that at the studies, at the end of the studies, at the levels that we were getting exposed to, using very conservative statistical analysis there, we weren't seeing effects that we would consider, what classical toxicology on that. We did at the higher doses, and of course, again, this was across 15 different groups that were looking at BPA, and so. We all know that these issues are out there. We're now moving into looking at PFAS. That's a very and the forever chemical is a very serious issue right now. And our group in bioinformatics and biochemical in biostatistics, we're doing some modeling as well, looking at food products or any type of FDA-regulated products, and seeing if we can use machine learning methods to predict toxicity, from these compounds. And so I think I'll go ahead and close it with that, because I know we've got some questions coming up that I'm sure people are raring to ask.
Excellent. Thank you so much, Dr. Patterson. It's great to hear perspective from FDA and what you're working on over there. Absolutely right. We do have some questions in the chat. We'll start with this one for Catherine Opie. Are there any updates on research by the EPA on nanotechnology in menstruation products, among other products people are using, like makeup?
Thank you for that question. I don't have specific updates for those types of products, but I can share that this is definitely an area of active work at EPA, both in our research and our regulation sides. Our Office of Research and Development has a lot of work going on with nanomaterials. There's also an issue paper that you can find by just browsing the website. Just look for EPA Nano. And then nanomaterials are regulated under our Toxic Substances Control Act. So there's a lot going on there, including, I think, some proposed rules, information requests. So the short answer is yes. I don't have anything specific for those types of products, but there's definitely work ongoing.
Can I chime in really quick, Maggie, on that? Because we have done some work in this area, especially with feminine hygiene products, and looking at compounds that are used, especially some of the silver compounds that are used for holding down microbial growth, and examining those again with our Office of Women's Health at FDA, and seeing the ability of these compounds to cross the vaginal wall and actually get into the body. And so we have published reports on that.
Excellent. Are those reports available to the public?
Yes. Mm-hmm.
Excellent. Could they find them on the FDA website?
You can probably just find those in PubMed if you just do a NCTR, and same way with the nanotechnology, but it is on the FDA website as well under our nano work.
Excellent. Thank you. Great. So our next question was also posed during Catherine Opie's presentation, but I invite any of the panelists to address it. What can consumers or the general public do if there are concerns about Superfund sites in their area with EDCs and there are health concerns in the community?
Another great question. So the first thing I would recommend is, know what's around you. So EPA offers a tool that you can search from our website, just plug in your zip code or something and see what's in the area, see what the chemicals of concern are. If you have specific concerns, our regional offices actually have some contacts, and I'm going to drop this link in the chat, if I may, as well. You can call our regional office, the one specific to your location, and ask to speak to somebody in the Superfund Community Involvement Program, and they can connect you with more information. Thank you.
Yeah, and can I also add to that, because I have quite a bit of engagement with the Superfund, either the programs, the centers themselves, or their program officers, and they are very responsive. So if you have a question, look to see if you have a Superfund center in your area, and you can find those by Googling it. If they come up, they pop right up, and contact the lead from the center. They may be able to answer your questions directly. I find them to be incredibly responsive, and they want to engage with the community. So, and know what the community feels like the problems are. So I would try them first and then do what Catherine said, because I think, they really want to hear from the communities.
Great. Thank you, Dr. Sue. This next question was posed during Dr. Patterson's presentation. Is DES included in the estrogenic database?
Yes, I believe it is. Yes.
Great. Very simple. Love it. Great. We have another question here. Some concern on BPA's building up in the human body. Is that something any of your agencies are concerned about that they're working to address?
Well, when we've examined the bioaccumulation as far as the pharmacokinetics of BPA, it's pretty rapidly and extensively conjugated and excreted. Most of our, all of our toxicokinetic studies were done using deuterated BPA, and so again, we're ignoring maybe what might be ubiquitous, and so we're monitoring the deuterated BPA that was administered to the animals, and then we can collect what's excreted and do our calculations to see if there's bioaccumulation there, and also looking at various tissues. And we really don't see a bioaccumulation, or at least not with bisphenol A.
Thank you, Dr. Patterson. I have another question for you. ECHA has recently reached the decision based on the totality of evidence that BPA restriction levels were not protective and dropped them significantly. Is FDA reconsidering any statements on BPA, or are you able to speak to that?
Well, I mean, I can say that we always look at the totality of evidence. I mean, when new data comes out, we're always interested in looking at that. And I know Dr. Fenton and her involvement with the NTP, I mean, we're always, assessing, new data that comes out to see if, our stance is going to be altered and what our recommendations are.
Thank you.
But I can't speak for the regulators.
Of course, understand for one part of FDA anyway. Similarly for this question, if this is part of your area of expertise, if you're able to answer, what is FDA's reaction to the low-dose BPA findings and clarity as published by the academic researchers?
This is an area of contention, of course. When you're trained in toxicology, we all know that Paracelsus says the dose makes the poison, and when you see U-shaped dose response curves, they really don't make sense for a toxicologist. And so some of the issues initially were statistical findings. I mean, we were very conservative with our statistics and how everything was assessed, and the low dose but causing an issue, but the medium doses don't, but then the high doses do. It really just contradicts what we've seen with other compounds, and so it just doesn't satisfy your the standard toxicology study.
Great, thank you for that answer. And I think we have time for maybe one more question. Is there a known risk exposure for using eyeglass lenses made of polycarbonate material for any of the panelists?
But I would like to address the other question about the PFAS and breastfeeding that Sky Wheeler noted. We have recently done a systematic review of all the literature on PFAS and lactation duration, and it was published in Toxics a couple of months ago, and there is evidence showing that PFAS will shorten lactation duration in women. There's an association, and there's been another recent paper out by Megan Romano showing that also. Just to answer that question.
Thank you, Dr. Sue. That's unfortunately about all we have time for. We head over to our next presentation. So thank you so much to all of the panelists for your excellent presentations and taking the time to be here today, as well as thank you for the people asking really insightful questions. We're happy to answer, and thank you for taking the time to be here as well. So our next presentation is from Christine Papagni on state interventions. Christine, over to you.
Great. Thank you so much. Am I echoing?
No, you sound fine to me.
Okay, great. Just want to make sure. I've been really encouraged by today, this two-day symposium. It's been great to hear all of the research that's going on, some of the new resources that people are working on, as well as what some of the other government agencies are doing. And also I want to apologize if my voice goes out. I'm recovering from a bit of a respiratory virus. So hopefully I can, hang in there. All right, so I'm going to talk to you today a little bit about the California Safer Consumer Products Program, who we are, what we do, how we do it, and some of the products we're looking at that contain endocrine disrupting compounds. I can't seem to progress the slides, so if someone could do it for me. Giving you flight control, great. Thank you, Sophia. And I don't know why, but it's still not working. Oh, there we go. Okay. So for those who don't know, this California Safer Consumer Products Program is part of the Department of Toxic Substances Control, which is one of several department boards and agencies under the umbrella of the California Environmental Protection Agency. Oops, sorry about that. Okay. And then the Safer Consumer Products Program is one of 3 of our core programs, which we now kind of describe as the past, present, and future of harmful chemicals. We have our site mitigation program, which manages the cleanup of sites that have been either historically or currently contaminated with hazardous chemicals. We also have our hazardous waste management branch or program, which does exactly what it sounds like. It manages hazardous waste within the state of California. And then we have our fledgling Safer Consumer Products Program, which has been around for about 10 years now. And we are a small group of scientists and engineers, as well as some economists and other analysts who evaluate, synthesize, and summarize the great work of other scientists, such as those who have been presenting at this symposium, to evaluate, whether chemicals and products are potentially cause adverse impacts to the human health or the environment. And then we try to encourage or motivate through regulation for manufacturers to replace harmful chemicals with either safer chemical substitutes or possibly safer manufacturing processes. So the Safer Consumer Products Program came out as a result of the 2008 California Green Chemistry Law, which was part of California's Green Chemistry Initiative. This was actually the first green chemistry law in the nation of the US. And this law basically required the Department of Toxic Substances Control to create a process to identify and prioritize chemicals in consumer products that could be harmful, and also a process to analyze alternatives to existing hazardous chemicals. And in addition to the Safer Consumer Products regulations, we also enacted what are called the Green Chemistry Hazard Trait Regulations, which essentially define the hazard traits and endpoints that we evaluate when we evaluate the safety of chemicals and products. So the Green Chemistry Law has multiple objectives, not just to develop the process to prioritize chemicals and products, and also to evaluate the alternatives to hazardous chemicals, which I've already said, but the main point is to avoid regrettable substitutes and what many people call sort of the whack-a-mole method, when legislature bans a compound and then it gets replaced by an equally hazardous chemical or also of concern. We also want to basically incentivize manufacturers to search for safer alternatives. And we try to do this in a transparent way based on science-based decisions. And we like our process and regulations to be enforceable. So how do we do this? We have what we call a 4-step process. We have what's called a candidate chemicals list, which is essentially the menu of chemicals from which we can evaluate. And this was created basically by a list of 23 lists from other authoritative organizations who have already evaluated the safety or concern of certain chemicals that are put in consumer products. And then from that list, we evaluate those chemicals within the context of a product. This essentially takes a lot of time and energy and research, evaluating the research of other people and coming to decisions on the potential adverse impacts or harms for those chemicals in the product. And then through rulemaking, we basically establish what we call priority products, which are a chemical product combination. And after rulemaking, manufacturers of those priority products are required to evaluate those products for either, like I already said, for safer chemical alternatives or possibly to create a safer manufacturing process, which could minimize those chemicals, especially if they're contaminants. And then following an alternatives analysis, we have several regulatory response actions we can take. So our process is not necessarily to ban these chemicals in products. Like I said, it's to try to get folks to replace them, hopefully, with safer alternatives. But we have a list of different responses we can take. We can either require manufacturers to provide additional information to DTSC, or we could ask them to provide additional information to consumers, either through labeling, information on their website. We could also basically require them to do some what we call green chemistry research, especially if there simply aren't safer, feasible alternatives to a chemical in a product. We may require them to actually look for something that might be different, or we might require them to fund research in other areas that may result in safer products. So the scope of the products that we can look at, I explained already that the candidate chemicals list is what we call sort of our menu of chemicals we can look at. Every 3 years, we draft a priority product work plan, which we consider kind of our menu of products that we can look at within a 3-year time frame. And then within the purview of our authority, there are some exclusions, which include pesticides, prescription drugs, radioactive chemicals, and natural toxins. And we currently have a new work plan under development, and we'll be making that public soon, and also asking for public input. So stay tuned. If you pay attention to our Safer Consumer Products website, some information will be up about that shortly. So our current work plan has it has 6 different priority product, potential priority product categories, which include beauty, personal care, and hygiene products, building products and materials used in construction, motor vehicle tires, food packaging, cleaning products, and children products. So now I just want to talk a little bit about some of the product categories that we're either currently or already have evaluated that contain endocrine disrupting compounds. Nail products, while toluene and MMA are not considered endocrine disruptors, those are 2 product categories that have either already been established as a priority product, which nail products containing toluene has, and soon nail products containing methyl methacrylate will also be going through rulemaking. And we're additionally looking at other chemicals in nail products. For example, we've already proposed to regulate triphenyl phosphate in nail products, and depending on the literature, there is the potential for triphenyl phosphate to be an endocrine disrupting compound. And we've also initiated some original research, we did a study of about 150 different nail products and measured them for a variety of different chemicals and detected a few endocrine disruptors within them: dibutyl phthalate, diisobutyl phthalate, and I already mentioned triphenyl phosphate. Additionally, we conducted an information call-in where we reached out to over 200 manufacturers and requested them to provide information to us on the ingredients in their products. And that information is actually on our website. We've also been looking at hair straightening products, which contain a few different potential endocrine disruptors, including parabens, phthalates, and BP3. And we're also looking at certain leave-on products, such as moisturizers and other types of leave-on products, which contain parabens. We've also been conducting some research on menstrual products, which I already heard a question about that to one of the previous speakers. And those contain a variety of different contaminants, including phthalates, parabens, and bisphenol. Additionally, some future research is coming up. We've just started screening children's products. While it's partly related to women's health, because half the population of children are female or identify as female potentially. So children's products have been shown to have varieties of phthalates as well as parabens and bisphenol. And California, in addition to the Safer Consumer Products Program, there's a few other efforts to manage endocrine disrupting compounds in California and hopefully remove them from products. The Cosmetic Toxic Free Cosmetics Act was enacted, and as of 2025, there are essentially 24 different compounds that are going to be banned in all cosmetic and personal care products in California, including certain parabens, phthalates, and some 12 different PFAS compounds. And additionally, the Cosmetics, Fragrance, and Flavor Ingredient Right to Know Act requires that companies that make products that contain fragrances or flavors report those ingredients to the California Department of Public Health, to their Safer Cosmetics program. And while they don't necessarily put those on the ingredient labels, they do share those on the website of the California Safe Cosmetics program. And then there's also a bill passed as of, I think it's 2022, professional personal care products that are sold in California must show the ingredients on the label. All right, so in conclusion, I just want to say that California is trying to address endocrine disrupting compounds and products in multiple ways. We've been asking manufacturers to look for safer alternatives, and some manufacturers are being proactive about this, especially when they see our priority product work plan. Or we also have a timeline on our website, which essentially shows what research we're currently working on. And so some manufacturers are being proactive even before their product may be regulated, which is great. And within our priority product work plan, we always have certain what we call policy priorities, and the health of women and children is a priority to us, and we give that special consideration. We continue to do research on different chemicals of concern in beauty, personal care, children, and then other products with endocrine disrupting compounds. So with that, here's my contact information if you'd like to reach out and have questions or want additional information. As well, you can learn more about California's Safer Consumer Products Program by joining our e-listserv, and you can get updates. And you can also learn about career opportunities as we are trying to grow our program. We're about to double in size, which is great because, you know, we are trying to do more, but we're a very small program. So again, I really appreciate the opportunity to be here today and speak to you all.
Thank you so much, Christine, for your presentation, for your work on the Safer Consumer Products Program at the California EPA. We do have some questions in the chat, so we'll start with this first one. Could you speak about the concerns regarding menstrual underwear? How would you compare the risk of menstrual underwear to tampons and pads, etc.?
Sure, and that's a great question. Currently, we have a small group of scientists who are looking at disposable tampons and pads, and potentially we'll look at the menstrual underwear in the future. We know that menstrual underwear have been shown to have PFAS compounds in them. In terms of comparing the risk, I don't have enough information today to actually share that with you. I apologize. But if you want to contact me, I can have you follow up with the group who's actually doing that research.
Thank you so much. The next question is on the restrictions on PFAS in California consumer products. Does it include fruits and vegetables or more makeup or food containers?
Okay, I can try to answer that question. We also have a staff person who is an actual expert in PFAS, so I can connect you to her because she could give you more information. As far as I'm aware, they're not being restricted in fruits and vegetables, but there has been a recent bill that passed that's going to restrict PFAS in food packaging.
Thank you.
Sure.
Okay, I think we have one or two more questions. Are you able to speak to the incontinence underwear products? Is that similar to menstrual, or is that under a different category?
It may be being looked at under menstrual products, under the disposable tampons and pads. I know the group did look at incontinence products, but since it technically is a different category, we have certain restrictions, not restrictions, but certain ways that we have to narrow the scope when we do our research. And so incontinence underwear would not fall within disposable tampons and pads, but that could end up, as we do the research, it could end up being a different product category that we could potentially regulate.
Thank you so much. So we are unfortunately out of time, but I do encourage anyone with other questions to contact Christine or to reach out to California EPA's website for more information. Christine, thank you so much for your time, and thank you everyone for the really insightful questions, really digging into these issues. We know that EDCs are of great importance and interest for people across a range of life experiences in society, and so we thank you for being here with us today. At this point, I will turn it over to Adrienne Smith, who is the Director of the Division of Policy and Performance Management at the Office on Women's Health. All right, give us 1 second. Thank you for your patience. Unfortunately, Adrienne is not able to give our closing remarks.
No.
Oh, my apologies. Here she is.
Sorry about that. Small technical challenges. Hello, everyone. I'm Adrienne Smith, Dr. Adrienne Smith with the Office on Women's Health, the Director of the Division of Policy and Performance Management. On behalf of our office within the U.S. Department of Health and Human Services, I would like to thank you for your participation over the past 2 days of the Endocrine Disrupting Chemicals in Women's Health Symposium. We covered a lot of ground related to this topic. heard from distinguished researchers and scientists, prominent advocates and champions, and in some cases, those with personal testimonies. We heard from women's service providers, federal program leaders, and respected organizations in communities, especially our challenge winners who have demonstrated successful innovative strategies to address gaps in knowledge and provide solutions to reduce exposure and risk for African American women. Across all categories of panelists and participants, we applaud you for being leaders and champions, working intently to contribute to the field of study and to inform the public about the impact of endocrine disrupting chemicals in health outcomes. The presentations and discussions were insightful, informative, and valuable to the work of the Department of Health and Human Services, as mentioned by the Assistant Secretary for Health this morning. Over the past 2 days, we learned what endocrine disrupting chemicals are and expanded our knowledge of their origin, and that they may be found in the most unexpected places. And there's science behind this. These chemicals have short and long-term consequences, which may have an impact cross-generationally. We also learned that there are small changes, safe, simple adjustments that can be made that can make a difference by paying attention to the things we put on, in, and around our bodies, as well as factors in our environments, home, work, communities. And we learned that outreach, voice, amplification of these messages and the information is important. We can expand our knowledge base around this topic and share with others through social platforms. Let's keep the conversation going. In the coming days, all registered participants will receive a post-symposium survey. We ask you to complete and submit the survey. Please take a minute and let us know what you have learned over the past 2 days, and also ways we can amplify messages and strategically plan future activities related to endocrine disrupting chemicals. We also invite you to follow us on social media, sign up to join our email list, and stay plugged into our work. Once again, thank you for being part of this pioneering and fascinating symposium. Have a great rest of your day.
Produced by the U.S. Department of Health and Human Services.